Hey, everybody. I just want to give this short little review of what we discussed so far with photosynthesis. Keep in mind that photosynthesis, once again, is all about energy transformations. This is why we go right into this unit after thermodynamics. Remember that thermodynamics is the study of energy transformations. So, with photosynthesis, we're looking at one specific type of energy transformation. And that energy transformation for us right now is the transformation of radiant solar energy into potential chemical energy. Okay, so let's just go ahead and start with the basics. Let's start with that equation and then go from there. So we start off, once again, we're dealing with carbon dioxide and water with the help of sunlight, um, basically taking those ingredients, carbon dioxide and water, and transforming them into potential chemical energy in the form of glucose. And then we get a nice little waste product out of that as well, oxygen gas that we breathe. So photosynthesis is a study of energy transformation. It is a part of thermodynamics. We're studying how radiant solar energy is transformed into chemical energy, glucose. Right, so this is all we're really basically talking about is that process, how that happens, how we can transform something like sunlight into glucose. And it's all about these molecular interactions. Right? So let's just start from the beginning. All right. So photons of light from the sun. Remember that photon is just a little packet of light energy. Photons of light from the sun are constantly cascading over everything they interact with. One thing they always interact with, of course, are the leaves on plants. And so if we have our leaf right here, we can actually peel down the layers, peel back the layers, and look what exactly is happening here. So radiant light hits the surface of a leaf. Those photons of light are going to travel through the cuticle, they're going to travel through the upper epidermis, and they're going to be captured. They're going to be captured by structures inside the palisade mesophyll. Remember that the palisade mesophyll layer of a leaf is packed with all these cells that contain chloroplasts. Chloroplasts, of course, contain the pigment molecule chlorophyll, among other pigment molecules as well, but primarily chlorophyll. Pigments are just molecules that absorb light. So what's happening here, the radiant solar energy falls over the leaf. It's going to go through the leaf down to the palisade mesophyll, where it's going to encounter cells that have chloroplasts. The chloroplasts are going to be able to capture that radiant energy and transform it. So a chloroplast itself is not a standalone structure. We know this. We know that they actually contain these other structures inside of them. Those structures are called thylakoids. So everything that we're going to talk about, all these different reactions are actually happening in, on, and around the thylakoid membrane. In, on, and around the thylakoid membrane. I've got the membrane marked here by these two little tick marks here. Right? So everything inside the thylakoid is considered the lumen. The lumen, that's that hollow section inside the thylakoid. Everything on the outside of the thylakoid, so still inside the chloroplast, but outside the thylakoid, that's the stroma. Right, so everything that we talk about with photosynthesis will take place either on the membrane, in the lumen, or in the stroma. Right? So starting things off with photosynthesis, we need to start with the light reactions. Okay, the light reactions. Those reactions that depend upon light in order to happen. So with this part of the photosynthesis equation, all we're really dealing with right now is water, sunlight, and oxygen. The carbon dioxide and the glucose will come later in what's called the light independent reactions. Right now, we just care about the light reactions. What's happening with the sunlight? So we go back and we talked about how these photons are striking the leaf, they're penetrating the leaf, and they're going down. all the way to the chloroplast, and inside the chloroplast to these pigment molecules that are located on the thylakoid membrane. So on the thylakoid membrane, we actually have other structures. Okay, so we're just dealing, once again, right now, we're just dealing with what's on the membrane of the thylakoid. Okay, the membrane of the thylakoid. And on that membrane, we actually have what's called a photosystem. We have a photosystem. This one is photosystem 2. We start with photosystem 2, PS2. Photosystem 2 is really a cluster of harvester pigments. Harvester pigments, I'm just going to indicate with these little circles here. Harvester pigments and what's called a reaction center. This one is P680. It gets that name, just means pigment 680. It's absorbing wavelengths of light that are 680 nanometers. Don't worry about that too much, but just know that photosystem 2 contains a reaction center that's called P680. Photosystem 2 also contains these harvester pigments. The vast majority of these harvester pigments, if this is a green plant, the vast majority of these harvester pigments are going to be chlorophyll. There are, however, other pigments, what are called accessory pigments here as well. It might be something like xanthophyll or carotene. All right, so you might have other harvester pigments there as well, but for green plants, the primary, the most abundant pigment is going to be chlorophyll. So this whole thing right here, this photosystem 2, this structure, photosystem 2, where you have harvester pigments and a reaction center, that whole thing together is called the antenna array. Okay, so antenna array. And as we've already mentioned, we have light, photons of light cascading all over this leaf and being captured by the structures on the thylakoid, right? Thylakoid membrane. One of these structures is photosystem 2 with all these harvester pigments. Pigment, once again, is just a molecule that reflects or absorbs light. So here we have these harvester pigments. We got these photons flowing in, and the photons are actually going to strike photosystem 2, and their energy, the energy of these photons is going to bounce around from harvester pigment to harvester pigment. And eventually the energy from these photons will reach the reaction center, P680. Photons of light carrying this energy, bouncing around from harvester pigment to harvester pigment, eventually reaching the reaction center, P680. And what's going to happen here is all this energy gets transformed. It gets transformed into P680. And we already know from the chemistry unit way back when energy encounters atoms, electrons can become excited. So what happens here with photosystem 2 is one electron becomes so excited it actually jumps energy levels and gets taken away. So energy is transformed to energy gets transformed to P680. P680, the electron in there gets so, or one of the electrons in there gets so excited, it jumps up to another energy level and gets taken away. It's taken away by something called a primary electron acceptor, PEA, the P. Okay. Electron gets taken away by a primary electron acceptor. I'm going to have E with a negative sign just to denote that's an electron getting taken away by the P. This electron is going to end up going on a ride. Right. This electron is going to go on a ride. The ride it goes on is called the electron transport chain. The ride it goes on is the electron transport chain. Okay, so this is the electron going on this little ride. Now, we've taken an electron from P680. We've taken an electron from the reaction center. It has to be replaced. All right, it can't just be down one electron every single time, light cascades across a plant leaf. So that electron is going to have to be replaced. And this electron will be replaced by a process called photolysis. Photolysis, right over here. Photolysis. Remember, photo just means light. Right? Lysis means to split or to break. So this is photolysis is using light to break something, to split something. And we're going to split something specifically in the photosynthesis equation. We are going to split the water molecule. That's how we're going to replace this electron. We're going to split the water molecule. So with photolysis, what's happening is water is being split. Water gets split into hydrogen ions, electrons, and oxygen gas. So water gets split into hydrogen ions, electrons, and oxygen gas. So this electron right here from photolysis, this electron is going to go over to photosystem 2. It's going to go up to the reaction center, and it's going to replace the electron that was lost. Remember, this electron got lost. It was taken away by the primary electron acceptor. We need to replace it. It gets replaced due to the process of photolysis. So this is photosystem 2. Like everything that happens in the light reactions with photosystem 2, this is it. If you are asked on a quiz, which you may already have been or will be asked soon, to draw photosystem 2, this is what we're looking for. We're looking for the diagram labeling photosystem 2. We're looking for this antenna array containing harvester pigments and the reaction center. We're looking for the understanding that due to the transfer of energy from harvester pigment to harvester pigment, the reaction center from photosystem 2 actually loses an electron, gets taken away by a primary electron acceptor, goes on the electron transport chain. And finally, we're looking for the idea that you understand that electron has to be replaced. And that electron is replaced through the process of photolysis. So this is part of the light reactions of photosynthesis. If you want to pause it here, just to go over, this is what you need to know for photosystem 2, the light reactions. This is the whole thing. I'm going to go on and talk about photosystem 1 as well as the electron transport chain in just a second. But if you want to pause it here and practice this drawing, now would be a good time to do so. I'm going to pause myself real quick. Okay, so we are back now. I just want to now talk about the electron transport chain and what's happening over in photosystem 1. Okay. So we know that our electron is being taken away by this primary electron acceptor, and it's going on the electron transport chain. So now we want to talk about, well, what is the electron transport chain? The electron transport chain is a series of oxidation reduction reactions. Oxidation reduction reactions. Those are also called redox reactions. Right, so an oxidation reduction reaction and a redox reaction are the same thing. And all we're talking about with redox reactions are molecules either gaining or losing an electron. Molecules gaining or losing an electron. That is a redox reaction. So this electron transport chain contains a series of redox reactions. You've got this electron here, the electron that came from the reaction center of PS2. This electron is being transferred from molecule to molecule. And so as one molecule gains that electron, it's being reduced. Right? So reduction in this case refers to a reduction in charge. Remember that electrons have a negative charge. So if you take on an extra negative charge, your overall charge is going to be reduced. So in this case, each molecule that receives this electron as it's being transferred from one to the next is being reduced. The ones that give away the electron, the ones that lose that electron, are oxidized. Okay, so it's an oxidation reduction reaction. So you can always remember the acronym oil rig, right? Oxidation is loss. So if something loses the electron, it's oxidized. And then reduction is gain. Anything that actually gains the electron has been reduced. So oil rig is one way to remember this. So this electron that's gone on this little journey is going to eventually be passed on. It's going to eventually be passed on to photosystem 1. And you might be asking, well, why does it need to replace photosystem 1? Well, that's a good question. So let's start off with, once again, we're going to have another antenna array for photosystem 1. Right? So this is PS1. And this time it's not P680 that's in here, it's P700. So pigment 700, wavelengths of light of 700 nanometers are being absorbed. Okay. So we once again have another antenna array. We have another reaction center, which means light is not only hitting PS2, it's also hitting PS1. So PS1 also has this transformation of energy, jumping from harvester pigment to harvester pigment, until it too reaches the reaction center. So the same thing that was happening in PS2 is happening simultaneously in PS1. And you already know what that means. You know that once this energy is going to hit the reaction center, once the energy from this hits P700, there's going to be yet another electron taken away. You already know that. It's going to be taken away by another primary electron acceptor. Okay? So we've lost an electron here. We've lost an electron from PS1 now. It gets replaced by the electron that came over from PS2. All right? So the PS2 electron that went on this electron transport chain ride gets transported to the reaction center of PS1, so it can replace the electron that was lost from its reaction center. This electron is also going to go on its own electron transport chain. Okay? So this electron is just constantly being moved around here. This electron from PS2 comes over to replace the one that's lost from PS1. And remember, the way that PS2 replaces its lost electron is through photolysis. So we're definitely replacing these lost electrons. We don't want some photosystem just constantly losing electrons and not being able to replace them. If that happens, then the whole thing just breaks down. So we've got this electron transport chain here. We've got another one right here. And one kind of cool thing about these electron transport chains is that the action, these redox reactions that are occurring actually generate their own energy. So what's happening is, as these redox reactions are occurring, we're actually going to have active transport take place. Let me back up for just a second. So everything so far, these photosystems that we're talking about, everything that we're talking about here has been right here along the thylakoid membrane, right on the membrane itself. Well, we're going to need something else happening. We're going to need more than just this in order for the actual energy transformation, the creation of glucose to take place. We need more than this. So what we're going to do is the energy that's given off through these electron transport chains is actually going to end up pulling down hydrogen ions. It's going to pull down hydrogen ions from the stroma into the lumen of the thylakoid. So these two electron transport chains, these series of redox reactions, are going to end up causing energy, enough energy to pass to actually pull hydrogen ions into the lumen of the thylakoid. Right? So energy from electron transport chains are pulling hydrogen ions into the lumen. Now, let's pause for just a second here, because when we said with photolysis, we were actually taking a water molecule and breaking it down into hydrogen ions, electrons, and oxygen. So we're getting hydrogen ions here as well from photolysis. We're getting hydrogen ions from photolysis, and we're getting more hydrogen ions from the energy being passed by the electron transport chain. So we're just constantly pulling hydrogen ions into the lumen of the thylakoid. So we're just building up hydrogen ions, hydrogen ions, hydrogen ions. Keep in mind, as a side note, a hydrogen ion is just a proton. Okay. So we got all of these protons, we got all these hydrogen ions just building up in the lumen of the thylakoid, in this hollow space, the opening of the thylakoid. Well, we already know from a previous unit that when we have a high concentration of something in one area, it's going to eventually want to diffuse, right? Things tend to go from areas of high concentration to areas of low concentration, right? Diffusion. So we're building up all these hydrogen ions in here. They are eventually going to have to diffuse out. And what's really kind of cool about this is they are going to diffuse through a molecule embedded in the thylakoid membrane called ATP synthase. Right? So ATP synthase. Hydrogen ions that have been pulled from the stroma into the lumen, along with the hydrogen ions that are generated from photolysis, are all building up in the lumen, and they will eventually pass through ATP synthase. So the passing through of these hydrogen ions through ATP synthase has its own name. It's called chemiosmosis. Chemiosmosis. That's what this is. These hydrogen ions that are built up in the lumen passing through ATP synthase during photosynthesis. Chemiosmosis. And the energy generated from these hydrogen ions passing through ATP synthase, that is going to give us ATP. All right? It's going to give us ATP. ATP will be used in the light-independent reaction. It's going to be used in what's called the Calvin cycle. ATP is going to be used in the Calvin cycle. So this action of these hydrogen ions passing through ATP synthase is chemiosmosis. The action by which ADP is made into ATP, the action by which that phosphorus, that extra phosphorus, is attached onto ADP, thus making ATP, that has its own name. So ADP, adenosine diphosphate, plus another phosphate, giving us ATP, that has its own name. It's called photophosphorylation. Photophosphorylation. So I'm just letting you know, because you might see that word again in the future. I just want you to know that's what that is. It's just the action of taking a phosphorus and attaching it to an ADP. When you take one phosphorus and attach it to ADP, you create ATP, adenosine triphosphate. In this case, it's called photophosphorylation. When we get to cellular respiration, next unit, you're going to see a different name. It's going to be called oxidative phosphorylation. It's all because in this case, everything's driven by these light reactions. So photophosphorylation, phosphorylating something is just adding a phosphorus to it. Photophosphorylation. This is phosphorylation driven by light reactions. That's why this is happening. That's why that phosphorus is able to be attached to ADP. Okay, so we're almost done. We've got one last thing to cover here, and that is our friend, the electron. This electron, of course, that went on this electron transport chain ride. Well, where is it going to end up? That's the last thing we want to talk about. This electron that went on this electron transport chain from photosystem 1 is going to be taken away by another electron transport molecule. It's called ferredoxin. Ferredoxin is going to take this electron and it's going to attach it to something called NADP+, thus making it NADPH. Right. So this electron from the electron transport chain gets taken away by another electron carrier molecule. This one's called ferredoxin. Ferredoxin is going to take it out into the stroma. So we've left the thylakoid membrane. We're going out into the stroma. Ferredoxin is going to take it out to find something called NADP+. NADP+ is another energy molecule. We know that ATP is our primary energy molecule. The vast majority of chemical reactions that happen in the cell happen because of ATP. However, some things might require a little less energy, not as much given off, so they can use molecules like NADPH to do that. NADPH is like the dollar store version of ATP. Kind of does the same thing, but maybe it's not the same quality. Right. So NADPH is going to also go on to the Calvin cycle. All right. So this is what happens to our friend the electron down here. It's taken away by ferredoxin, out into the stroma, joins with NADP+ to make NADPH. It reduces NADP+ to make NADPH because the electron's been added to it. Just as an aside, if you're wondering how NADP+ has an H on the end later, well, all those hydrogens that are out in the stroma, some of them, of course, were brought down into the lumen to build up this chemical gradient to allow chemiosmosis to occur. But there are still some other hydrogens out there. So you have NADP+, it's merging with the hydrogen, and it's having this electron added to it. So remember over here with photolysis, we lost a hydrogen, we lost an electron. Now they're coming back together. The NADP+ becomes NADPH out in the stroma. So everything that we talked about, all these light-dependent reactions, everything that's occurring in here is all being done just to generate two compounds. All this stuff is happening to just make two things. The light reactions, what we really want out of this, what we really have to have, are ATP and NADPH. All right? These two molecules are going to drive the Calvin cycle. They will provide the energy necessary for the Calvin cycle to occur. If ATP and NADPH are not made in the light reactions, there is no Calvin cycle. If there's no Calvin cycle, there's no glucose. Those two energy molecules are going to be used to help convert that carbon dioxide into glucose. So that's why all this is even happening in the first place. Okay, so I know this is kind of a long video, but I just want to go through every single step in the process, just as a little background information here. Okay, this is all just light reactions. That's all we talk about are the light dependent reactions. That's it. Light independent reactions, the Calvin cycle, will be discussed in this upcoming week. Right. If you have any questions, clarifications, anything, please let me know. Otherwise, I hope this helps. Remember that your quiz, you're going to be asked to draw out photosystem 2. So just make sure you include these details here: photolysis, PS2, harvester pigments, reaction center, primary electron acceptor, electron transport chain. That's all for photosystem 2. The rest of it is just the rest of the reaction. What else is happening here? Okay. So if you have questions, let me know. But otherwise, I hope you have a great day, and I look forward to seeing you in class. Take care.